Okay, Lawrence, would you like to start or shall we wait a couple of more minutes?
We can start whenever you're ready.
Okay, I think let's get going and then more people can join in as they come. Okay, so a very warm African welcome to the first Biophysics in Africa conference. So for those of you who don't know me, I'm Chart Kruger from the University of Pretoria in South Africa, and I'm really excited about this event and to witness how many biophysics activities are already taking place in Africa so that we are able to organize a week-long conference. We have an amazing program of talks by both African and some very prominent non-African speakers, covering a very broad range of topics within the broad field of biophysics. And I would like to encourage you during this conference to broaden your horizons by getting introduced to topics you're not familiar with. For example, if you're from a physics or chemistry background, this is a valuable opportunity to learn more about the intriguing facets of biology. And if you're from a biology background, you can appreciate some of the rigor offered by physics. After all, cross-disciplinary research is the future. So this conference also displays some of the growing research excellence on the African continent. Africa has enormous capacity and talent, which has the potential to even outperform the Western world if nurtured and grown appropriately. By working together, we can use our wealth of resources to tackle local, continental, and global challenges. In light of this, I would like to encourage you to please join the open forum today and also on Friday at 12:30, where the African Physical Society and some of our other co-conveners will discuss the African strategy for physics and also some exciting research facilities and opportunities. So come and share your own ideas and listen to the ideas of others. If this is your first virtual conference, I'd like to welcome you to possibly the new norm. These virtual conferences aren't quite the same as in-person, face-to-face ones, but they do offer a lot of benefits, for which I'm really grateful. Right, before we start, there are a couple of housekeeping items I'd like to mention. So first of all, please keep yourself muted during a presentation and only unmute yourself when you are asked to speak. Then after each presentation, please use the raise your hand button if you'd like to ask a question. This button you can find at the bottom of the screen. It's grouped together with some other reaction icons. And we would prefer the question to be asked out loud, but if your mic doesn't work, you might obviously write your question in the chat. And then if you have more questions after the Q&A, you're welcome to continue in the chat. We have assigned 20 minutes to each talk, or to most of the talks, actually each talk today. So 15 minutes is assigned to the talk and 5 minutes to Q&A. So I will stop you after about 15 minutes. Or the other session chairs will also stop you when you have about 5 minutes left of the total 20-minute slot. Okay, you can find the latest updates on the conference website. For example, a very useful feature is the timetable, which you can click into and see further detail. Please note that all the talks will be live streamed to YouTube and will also be recorded and put on the YouTube channel of the African Light Source after the conference. If you do not give consent to this, for example, if some of your material is embargoed, please email the organizers and let us know about this. Finally, I'd like to thank all the organizers and co-convening organizations whose information you can see on the conference website. And then lastly, a great example of humility is a man whose name you have never seen on the conference website or in the emails, but he's in fact the main organizer of this conference who has been working behind the scenes. And this is Lawrence Norris. Lawrence, I hope you don't mind that I'm acknowledging you here, but I really wanted to do that. Okay, I've eaten a couple of minutes into the first session, so we'll move all these talks a few minutes forward. It's now 10 past 10, so let's start this first session, which is on quantum biology. And quantum biology is a relatively young, but really a fascinating research field and a very fast-growing research field. The first speaker of this session is Mr. Calvin Mpofu from the University of KwaZulu-Natal, who has very recently completed his PhD, and he will be talking about quantum plasmonic sensing. Right, Kelvin, the floor is yours.
All right, thank you. Good morning, everyone. My name is Kelvin Pof, and I'm going to be talking about quantum plasmonic sensing. So this is part of like work which I did with Professor Mark Tame, who is at Stellenbosch University, and yeah. Okay, so in my talk, I'm going to start off by introducing surface plasmon resonance, which is abbreviated as SPR, and the Kretschmann configuration. Then I'm going to present a theoretical sensing setup to model measurement noise for quantum states, which we'll be interested in looking at. And then I'll give a discussion of some results. And finally, I'll give a conclusion. So in a surface plasmon resonance experiment, you have light coming from a light source going through a glass prism and hitting some metal surface before it's reflected to some detector. So something interesting happens as you change the angle of incident theta here. What you observe is you see that as you change this angle, as you change the angle, the intensity that your detector picks up starts to drop towards a certain minimum, which we call the resonance dip. And after it hits this minimum, it starts to go back up. And the phenomenon that causes this resonance dip is called surface plasmon resonance. So here, the light from your light source couples with conduction electrons, which are inside the metal surface, this metal surface sheet. It's a very thin sheet, which is usually nanometers thick. And so, yeah. So when the light couples to these conduction electrons, it sort of generates like an evanescent wave, which propagates on the surface of this metal, which propagates on the metal surface. And as the coupling gets stronger between the light and these conduction electrons, you start to notice that your detector starts to pick up less light because less light is being reflected back. So this setup which I'm describing here is called the Kretschmann configuration. There are other configurations which you can use to excite these surface plasmons, but I won't discuss them here. This setup is very useful for binding experiments which extend themselves to biosensing applications. So our metal surface is usually coated with some biomolecule, which we call the receptor. So these small biomolecules are actually coatings on top of our metal surface. And yeah, typically in a biosensing experiment, so what you do is you allow a buffer to flow through your setup. So these will be your receptors, and the buffer just flows through some flow cell over your receptors. And typically this buffer is useful for setting the baseline for your experiment. And you'll notice that you'll still have your surface, you'll still see your surface plasmon resonance dip. And after you pass through your buffer, you pass through an analyte which contains the biomolecules that you want to investigate. And these biomolecules are called ligands, and these ligands tend to bind to the biomolecules on the surface of your gold, which are the receptors. So what you'll notice is when this binding action starts to happen, the resonance dip will start to shift. The position of the resonance dip will start to shift. And you can you can sort of like plot a time series of this shift in your resonance dip, and we call this a sensogram. So these sensograms are quite useful because they allow us to measure certain kinetic parameters, which are the Ks parameter, the Ka parameter, the Kd parameter. And so the Ka parameter is called the association constant. So it tells you how quickly your ligand binds to the receptor here. The Kd parameter is the dissociation constant, which tells you how quickly the ligand unbinds from your receptor. And the Ks parameter tells you how strong this binding reaction is. So these parameters are very useful, are very important, and they describe all the information that we need to understand the binding reaction. So this setup can be used for a wide, it has a wide range of applications, which include the drug discovery and vaccine design and diagnosis of viruses in clinical samples. So the question we wanted to address was, okay, I'll describe the issue really. So we know from metrology that when we take measurements using classical states, there's a limit imposed on the noise measurement, and this is it's called the standard quantum limit, or the short noise limit. And typically we can use coherence states to reach this limit. But we know that that we can use quantum states to go beyond this this sort of limit in precision towards another limit called the Heisenberg limit. So the question we had in doing this task was, can we use quantum states of light to improve the precision of the kinetic parameters which we extract from our sensograms? So yeah, this is more of a theoretical exercise, and so we had to model the measurement noise which we would get for different states if we were to use different states of light. So we used this reference, Lee et al. In Physics Review A, to get the general description of the noise that you would get for different quantum states. And the quantum states that we looked at were the coherent state, the pair Fock state, a two-mode squeezed vacuum state, and a two-mode squeezed display state. So this slide just shows how you would prepare these states, but I won't really get into detail on that. So our next task was to simulate an SPR experiment where, so initially what we have is some ideal sensogram which represents an experiment which we did. Then we add different, we add the noise which we calculated, which we modeled here, to each of these sensograms. So if you add the noise from the Fock states, from the coherent state, and this is separately, by the way, so from the Fock state, the coherent state, the two-mode squeeze vacuum, and the two-mode squeeze display state. Then we fit the now new sensogram, which you get once you add the noise. So say for instance, we had added noise from a coherent state measurement. This would be the new sensogram, and we would just fit this sense, whatever kinetic all those kinetic parameters that we're interested in. So we wanted to get statistics for these kinetic parameters so that we can see how they're distributed. And so we had to do some Monte Carlo simulations for these SPI experiments. And we also measured something which we called an enhancement, which was just a measure of the ratio of the standard deviation for the coherent state versus the standard deviation for all the other quantum states that we were looking at, which is the 2-mole squeeze vacuum, the 2-mole squeeze displaced, and the Fock states. So these are just the results that we got. So we were also interested in looking at this parameter called nu, which is the sampling frequency for our measurement. But in this talk, it's not really the primary thing that I wanted to focus on. So I wanted to point out the kinetic parameters. These are the kinetic parameters that we measure for all the different states. So we have the dissociation constant, the Ks parameter, and the association constant. But I'm going to focus on just one of these, one part of this whole picture, because the idea is similar throughout. So if you look at this diagram, this dashed gray line, that represents the real kinetic parameter that our system was supposed to estimate, So when we use 2-mole squeeze vacuum states as input, we see that we have this green line, this green point, which has a very large error bar. And when we use the 2-mole squeeze display state, which is another quantum state, we see that we get this blue error bar, but they're both still worse off than the black line, which is the coherent state. And that's sort of like, that's not what we were that's not what we were hoping for. We're hoping that the quantum states could perform better than the quantum states. And we also have, but we do see that one of the quantum states actually outperforms the coherent state, which is, this is the Fock state, this red point. And if we look at the enhancement coefficient, which we calculated, you notice that if the enhancement value is above 1, then that means the quantum state, if it's above 1, that means the quantum state performed better than the coherent state, and we were able to beat that short noise limit. And if it's less than 1, that means the quantum state that you're looking at was worse off than the coherent state, and we performed worse off than the short noise limit. So
Calvin, you have about 2 minutes left, so please take note of that and start to wrap up your talk.
Okay, thanks. So we looked at the photon number dependence as well, and basically it followed the same trend. And one thing that we did notice was that the 2-mole squeeze vacuum state performs worse as you increase the photon number. That was the one interesting thing that we saw out of this. So there was a case where if there's a situation where if you manipulate the noise in your system, you can get your 2-mole squeeze vacuum state and your 2-mole squeeze display state to outperform the coherent state, but only slightly, but it does outperform the coherent state. But it makes the Fock state, adding that noise into your system makes your Fock state perform a bit worse. So just to quickly kind of recap what I've been trying to say, or what I've been trying to explain, is we had a way of modeling. So we had a way of modeling the measurement noise we would get if we input different quantum states of light into our into our SPR setup. And so we started off by constructing, since this is an experimental exercise, we had to construct an ideal sensogram, then add this noise which we modeled, then fit the now new noisy sensogram and extract parameters from there. Then we compared the parameters which we extracted for the different states, and we found that in certain cases, the parameters that you get when you use quantum states of light perform better than the coherence state. And we can say that in general, using quantum states does bring advantages in certain advantages in certain cases. And in general, the pair Fock state seems to give the best advantage. Though I would like to note that whilst in theory, we do get this advantage by using quantum states, when you go to experiments, there's so many losses in experimental setups that this advantage is kind of lost as the efficiency of your setup goes down. Yeah. Thank you.
Okay, thank you very much, Calvin, for a very interesting talk. This is really a very interesting topic. So the floor is now open for questions.
Remember to unmute yourself when you're asking questions.
Yeah, or you can raise your hand. That might be easier for us to identify you. So if you go down to the reactions icon, there you can see a raise your hand button. Maybe to break the ice, Calvin, so theoretically the Heisenberg limit offers a root n improvement in your signal to noise ratio. So where n is essentially the number of parallel detectors in your experimental setup. But in practice, there are many sources of noise that make it very difficult to get any real improvement to the standard quantum limit, like you have also mentioned here in your last slide. Are you able to give a ballpark figure to estimate approximately how much do you think is a realistic improvement that you'll be able to obtain if the experiment is done really well?
You mean in my specific experiment, or in general?
Well, say, so I've come across this one Chinese group that has used 16 detectors. So in principle, they would be able to get a 4 times improvement. But in practice, you get hardly any improvement because of all the sources of noise. So say you would be able to use 16 detectors. What improvement do you think you would really be able to get?
Yeah, that's a bit of a tricky question. I'm not sure, but we did an experiment in our lab where we use single photons, and we had so many losses that the best efficiency that we could get was around 20%. So so that was the best sort of like from our experiment, that was the best outcome we could get. Like we couldn't get any more than 20% output or efficiency from our setup. I don't know if that answers the question.
Yeah, definitely. And that was using only 2 detectors.
That was using only 2 detectors.
Okay, that's really good. That's really promising. Thank you, Calvin. So any other questions? So we do have time for maybe one short question. Okay, if not, you're welcome to type out some questions after the talk. We can continue to discuss with the presenter in this way. And then I'd like to give the presenter Calvin a round of applause. So thank you very much for a very interesting talk.
Oh, thank you. It was a bit. I think I threw in a lot into a short amount of time, so maybe I lost some people somewhere.
Yeah, don't worry. That always happens, and that is the idea of this conference, that we give everyone a flavor of what you are doing. If someone is more interested, then obviously they can get more information from you personally. So thank you very much for this, Calvin.
Okay, thank you.
Right, so the second speaker for today is Kimara Naiker, also from the University of KwaZulu-Natal. And she is still in the first phase of her PhD, if I understand it correctly. And she's going to talk about applications of machine learning techniques to the description of quantum coherent excitation energy transfer within the dimer model. Right, so you're welcome to share your screen, Kimara. And yeah, looking forward to your talk. The floor is yours.
Okay, cool. Can you see my screen and hear me clearly?
Yes.
Yes, I can.
Okay, cool. Just 1 second. Okay, cool. So let me just test that this is fine. Okay, so good morning, everyone. Yeah, my name is Kimara Naiker. I'm a PhD student in the Quantum Research Group at UKZN. And yeah, so I'm going to present some preliminary results from my PhD study so far. And it's, yeah, my topic is basically applications of machine learning, yeah, techniques to the description of, well, yeah, excitation energy transfer, and specifically in this two-level system or the dimer model. So starts off with photosynthesis. So we may all know this process, and it's where light energy is transferred to chemical energy, right? And this mainly happens in green plants and a few other, like certain organisms. So the one we're most interested in is green sulfur bacteria. And yeah, so in the first step of photosynthesis in this particular organism, energy is absorbed in these antenna pigments up here, and it's subsequently transferred down to the reaction center. And this is where photochemical reactions are triggered. And now between these two components is where the, it's called the Fenner-Matthews-Olsen, or the FMO complex. Yeah, this is where that FMO complex is found. And it's a protein complex that's made up of these like 3 subunits, and each consists of 7 bacteriochlorophyll molecules. So the FMO complex, it serves as basically a bridging energy wire of some sort, because it's tasked with transporting the sunlight energy harvested in the antenna chromosome down to the reaction center. And basically it represents an important model in photosynthetic excitation energy transfer. And it's been extensively studied experimentally and theoretically. And one of the main studies, this was like back in 2007, is when Engel and some of his collaborators investigated this FMO complex. And they succeeded in observing some kind of long-lasting quantum effects. And this provided direct evidence for long-lived electronic coherence. So quickly, the observed coherence, it lasts for timescales similar to the excitation energy transfer timescales. And this implied that the electronic excitations move coherently through the FMO protein rather than what was previously proposed, which was incoherent, like a kind of incoherent hopping motion. And basically, also to mention that the idea of quantum coherence playing a role in photosynthesis arose from these observations that some energy or electron transfer processes in bacterial and plant pigment protein complexes were efficient to an extent that basically exceeded explanation using only classical theory. And now there's some controversy. So explanations for this observed like long-lived coherence has evolved during the past decade specifically. And so while quantum coherence was first proposed as the origin, more and more, and I've labeled a few here, experimental and theoretical evidence has supported proposals that this long-lived phenomena may arise now from either vibrational or vibronic coherence. So that's the current consensus. And it's an ongoing academic debate. But for our specific study, basically this ever more complex of quantum biology, it's a motivation for us to solve these, I will mention it later, something called the strongly coupled hierarchical equations of motion. And so for our case, this mixture or current like controversy doesn't matter too much. And we will gauge much further and deeper on in this specific paper by Ishizaki and Fleming, which I will mention now. So one of the viable approaches to explore this long-lived quantum coherence and its interplay with the protein environment is through the reduced equation of motion. And in this approach, the key quantity of interest is the reduced density matrix. And typical situations in photosynthetic excitation energy transfer are such that electronic coupling strengths between chromophores and their local environment photons. A typical situation is when this coupling spans a similar range as the reorganization energies. And these reorganization energies that I speak about, they characterize the time scale of the coupled phonons relaxing to their respective equilibrium states. So in this regime now, these site-dependent reorganization energies, they cannot be described by theories that rely on the Markov approximation, because this requires the phonons to relax to their equilibrium states instantaneously, which basically means that the phonons will always be in equilibrium even under the electron-phonon interaction. So basically, we turn to this numerically exact formalism of quantum dynamics in this non-Markovian regime, which is called the hierarchical equations of motion. And yeah, as I previously mentioned, it was derived by Ishizaki and Fleming. And basically, the hierarchical equations of motion can describe quantum coherent wave-like motion and incoherent hopping in the same framework, and they reduce to these previously thought conventional theories in their respective limits of validity. However, a problem occurs where when we are needing to solve these hierarchical equations of motion now, it's computationally expensive due to the adverse scaling with the number of pigments. So before we get into that, I will just describe the hierarchical equations of motion further. So here we have the Hamiltonian, which basically energy transport in light harvesting complexes are basically modeled with this some kind of effective, in this case you can call it the Frenkel exciton Hamiltonian. And the total Hamiltonian is composed of the Hamiltonian of the system, the bath, and the system-bath interaction, right? So for the Hamiltonian of the system, we have epsilon j here, which is basically the excited electronic energy of the jade state. And J here is the electronic coupling and basically the transition amplitude between levels, and it's responsible for the excitation energy transfer between sites. Then we have the Hamiltonian of the bath, which is basically an infinite set of harmonic oscillators. And here, basically P and Q are your standard like momenta and position operators. And then we have the system and bath Hamiltonian. And basically here, I've summed it up into just G, this constant, which describes the strength of the interaction between each oscillator of the bath with the corresponding energy level of the system. And then now, so we have, as I mentioned before, basically the excitation energy transfer dynamics can be described by the reduced density matrix, right? And, or the reduced density operator, and that's embedded in the sigma operator here. So basically this, these, first I will mention these operators down here, phi and theta, are basically an operators, and they are, this theta specifically is actually an infinite series, but because we are working in the high temperature case, the series terminates after the first term, because high order terms are rather irrelevant in this regime. And basically working under these assumptions, one ends up with the sequence of coupled linear differential equations, which are known as the hierarchical equations of motion, highlighted in red here. And just to give more insight into what's actually happening here, I will just mention the two-level system. So here, this vector n, I mentioned it here on this slide. So basically, for the two-level system, here you can picture an actual hierarchy. So the hierarchical equations of motion can be broken down into these like equations further, or they expanded rather. So up here for the case where that vector n, where both n1 and n2 are equal to 0, is where you will pull out the reduced density matrix, which is of main interest to us. Then on the intermediate levels, you will have the auxiliary density matrices. And then on the highest level of the hierarchy, which is basically when the sum of the indices n, so n1 and n2, when they are summed up together, they will add up to 2, which is the highest level of our hierarchy. So for this specific case, you have like a terminating condition where the equation is specified by this equation here. Okay, so that's enough to mention about the two-level system. And then now we go into the machine learning side of things. So basically machine learning is like a part of artificial intelligence. And you have three main types. So for us, the main interest is supervised learning. So this is basically where the model learns which feature is associated with a label. So basically like a machine uses label data to train a neural network. And we want to derive the relationship between the input and the output without actually giving the model the law or the algorithm. And by this way, the model can learn to predict by being given enough input and output data, basically. So then you have also, just to mention, the unsupervised learning. And in this case, you basically have unlabeled data. So the model is trained to find patterns, and correlations between features by itself. And then reinforcement learning, which is basically reward-based learning, and it works on the principle of feedback. And yeah, as I mentioned, so basically how it works is that an input is fed into a model and an output is given out. And basically the performance of a model is gauged by how well the truth or the real output compares to the output predicted by the model. And just a few machine learning basics before I dive in deeper. So basically the most prominent and efficient architecture of machine learning are now called these neural networks. Yeah. So basically they form the basis of machine learning, and the structure is basically inspired by the human brain. It's also an extremely successful framework. So basically what happens here is it's made up of layers of neurons. So these blue dots are called neurons, and these are basically the core processing units of the network. And the input layer receives an input, of course. The output layer predicts the final answer, and the hidden layers here perform most of the computations done by the network. And just a little bit of behind the scene is the neurons are connected between layers through these channels, highlighted by these gray parts. And each channel is assigned a numerical value, which is called a weight. This is written as a W here. And basically the weights are summed up and input into the next layer. And each neuron has its own numerical value, which is known as the bias here, which is added to the input sum. So it may sound a little bit complicated, but I promise it's not. And basically the result is then fed into this activation function. So I've just shown a little skit here. And this determines whether that neuron will get activated or not. And only activated neurons transmit data into the next hidden layer. And this carries on. And basically in this manner, data is propagated feed forward through the neural network. And then at the output layer, the neuron with the highest value gets activated to give you your output. So it's all based on numbers, which is really nice. And basically the values can be seen as a probability. And yeah, so the predicted output is then compared with the actual output, as I mentioned before, and then the measure of error is calculated. And then this information is sent backwards through is that telling me I have 5 minutes?
Yes, you have 2 minutes left.
Okay, 2 minutes, that's cool. Okay, yeah, so that's backpropagation in a nutshell. I need to move a little bit faster. So our actual problem is now we have your Hamiltonian of the system with the environment also. And then this, I use basically what I call the hierarchical equations of motion solver, which is some Python code that once solved, it gives you your reduced density matrix and gives you some kind of observables, which is characterized from the reduced density matrix. And what I actually want to solve is the inverse problem. So basically, if you feed the neural network these observables, can it accurately give me the electronic couplings that characterize the system? Yes, that is a problem I'm trying to solve. And basically, I generated some data sets. So these were the ranges for the Hamiltonian parameters that I used. I used 10,000 samples for my data set. I split them into train, validation, and test sets. And basically, just to give you some insight, so to visualize the data set, these are the observables that I feed into my network, and then it will spit out the excitation, electronic coupling, and the intersite coupling as well. And basically now this is just a picture of my neural network. So I showed you a generic picture before, and this is mine. So I used a convolutional neural network, and these were the rough estimate of the total trainable parameters, number of total trainable parameters in the system. And basically I ran this is standard procedure to run some cross-validation on your model. So basically to make sure that your model is not like just happy to predict or performs well on a specific set of data. It like splits the data into different folds. I used 5 folds here to assure the reliability of the model rather. So these stats look pretty good. And then my results. So again, I mentioned this was for a 2-level system. So basically here you can picture that the green line here basically shows perfect agreement with the hierarchical equations of motion. And then the blue dots are what my model predicted. So I've put up some accuracy metrics here. And yeah, so we've got around 98, 99% accuracy. And this R-squared score is basically a statistical measure to show like how close the data are to the fitted regression line. And important thing to mention here is this data that I presented in the results are data points that the model has not seen during training or validation. So it's completely unseen testing data, which is important to mention. And basically, yeah, in conclusion, so we're happy with the way the model has performed on a 2-level system. So we would like to extend this further now to high-level systems and see how we can compare. Now, does the model need a little bit of tweaking? The architecture need tweaking? How does it perform there? And yeah, if it works well, we would like to extend this into an experimental setting. Now, well, in an experimental lab, you have real life observables, right? So if you throw this into my model, will it give you the parameters of the Hamiltonian? And by that way, you bypass solving the hierarchical equations of motion exclusively. You save time, memory, and yeah, a lot of computational power. So that is my study in a nutshell. So thank you.
Right, thank you very much, Kimara. Very interesting topic and very nicely presented. So
thank you.
So well done. The floor is now open for questions.
Yes, please.
Hey, Kimara. I'm Tuvan.
Yes. Hello.
I have 2 questions actually, but maybe I'll just ask one, and then if there's time, I'll ask another. So,
okay, cool.
Why did you use a convolutional neural network and not just a densely connected neural network?
Okay, sure. So of course, yes, I started off with even just the most basic, like some regression tree models, some multi-layer perceptrons, like the most simplest, and also just densely connected networks first. And basically the predictive power of them wasn't good enough. So I constantly use these 3 metrics of accuracy, the R2 score, and a mean squared error for loss. And I've tried many models. I started off, of course, with the most basic. And yeah, so I built up, and then I found, well, I tried out 1D convolutional first. It wasn't as good. And now I'm with the 2D convolutional neural network, which has the best predictive power. And that's why I'm sticking with it for now.
So the 2D network takes as input the figure basically, right?
Yeah, so
okay.
Yeah, so basically for the 2-level system, If I take the reduced density matrix, it's just a like 2 by 2 matrix, right? So I feed in from that reduced density matrix, I calculate two kinds of observables, which I just showed here briefly. I mentioned them as like sigma x, r, and I. So they can be calculated from the reduced density matrix. So I took two of those observables and I fed it into the neural network. So basically those are like my two features. Yeah, the network, yeah. And just briefly, these were basically like time series data, right? So like it showed like how these two observables were basically like just two arrays. And my time, my time that I calculated for was something like 5,000 femtoseconds, and it was calculated in just 1,000 steps. So that was like the shape of my input features, basically like 2 by 5,001.
Right, thank you for that very clear answer. Another question from the audience? Okay, TwoLevel, you said you had a second question as well.
I had a second question, but I forgot it now. But
you can
yeah,
always ask later, not a problem.
Okay.
I guess
okay. So that you remember, then you can go ahead.
Yeah, I just wanted to know the observables. So how would you measure the observable? Are they easily measured in the lab for FMO, for example? Or would you measure spectra, for instance, and then from the spectra try to determine the observables, and from the observables the energies?
Exactly that, yeah. So as far as I know, in an experimental setting, like a lot of 2D electronic spectroscopy has been used to basically observe these observables, and then that's how you get the data ideally to split into my model, yeah. So as far as I know, yeah, 2D electronic spectroscopy is one of the most popular techniques used.
Thank you.
Okay,
Welcome.
thank you. Yeah, you've actually taken my question, TwoLevel. Okay, so if you have more questions, please do make use of the chat box. You're welcome to continue to interact with the speaker. And yeah, so we won't hear you if you give an applause, but you're welcome to also use the virtual applause button in the reactions icon. So yeah, with that, thank you very much, Kimara. We're looking forward to see all the outcome of this research. Thank you very much.
Thank you.
Right, our next session, next speaker for this morning's session is Dr. Tao Li from the University of Nottingham. So we are very pleased to have him here with us, and we are very much looking forward to his presentation, which will be on the basis-independent coherence in avian-inspired quantum magnetic sensing. Also a very interesting topic. Right, Tao, you are welcome to share your screen. Right, we can see your screen. The floor is yours.
Okay, so my name is Tao, and I'm currently a postdoc at the University of Nottingham. And this work was done jointly with Alexandra Alai Castro at the University College London. So my topic is about basis-independent coherence in avian-inspired magnetic sensing. So there are a couple of terms here that I don't think that everyone is familiar with. So in particular, I'm going to talk a little bit about what I mean by magnetic sensing in birds, and then I'll go into what my simplified, like my quantum magnetic sensing model is. And I'll talk about what I mean by coherence, and in particular, what I mean by basis-independent coherence. So many migratory birds, as well as other animals, can use the Earth's magnetic field to orientate within it. Like, for example, there are behavioral studies where they put birds inside rooms and change the magnetic field and see whether or not they can react. But the mechanism of how this occurs is unknown. One possible mechanism that people have, like one possible candidate, is a light-sensitive radical pair mechanism. And this is where you have so two radical pairs recombining chemically, and then this chemical reaction is somehow detected by the animals, but it's not really known yet. But one possible molecule that can do this is called cryptochrome, and it's been found inside birds. and other animals, and it has like light sensitive and other properties, but people don't know. It's still like uncertain. But because it's like one of the main candidates, there's been a lot of work done with this radical pair sensing. So what happens is that these radical pairs, so these like molecules that have spins inside them that are in turn affected by the magnetic field, and that affects the chemical recombination, which is somehow sensed. And so this is like the main chemical reaction. You kind of have some singlet and triplet states, A and B, and they recombine and things happen. But that's not really important for my work because I work more on the quantum side. So in these kind of models, in the quantum models, they have particular elements. So you always have a Z interaction with the magnetic field. You have other factors that such as anisotropic G factors, hyperfine interactions with the surrounding nuclear spins inside the molecule, electron exchange interactions, and general environment interactions. So as a mini background, most models looking at the radical pair mechanism in quantum biology consider hyperfine interactions, or they consider the polar and electron exchange interactions. So in my work, they kind of differ from those because we also look at environment interactions instead of the previous ones. So our model contains this free radical. So we have C, which is we have the A and B, which is the main two radicals that combine chemically, and then you have the C, which is a scavenger, which helps, which is supposed to help the two original ones to react. And then you have some various interactions. So this is the model that we have in particular. You have A and B, which is the main two. We have C, which is the scavenger, and then we have the environment. And this is called a collisional environment. Basically, at each time step, you have a fresh environment state that interacts with the radicals, and then at the next step, they go away, and yet at each step over time, you get a new set of environments. So this is like literally the environment is colliding with the radical pairs at each time step. And there's some particular details about the interaction, but I'm not going to go into it. And this is basically the evolution equation. You have all the interactions that I put inside here. In this Hamiltonian, you have the state of the system and the environment over time, and you have this minus K factor, which is basically relating to the chemical recombination. So the important thing in the model that we choose is the collisional environment, because the collisional environment can simulate both Markovian and non-Markovian environments. So a bit like in Kamara's previous talk, she talked about how you had non-Markovian environments. So collisional environments is another way to simulate non-Markovian environments. And for the magnetic sensing in birds in particular, the viral form of the environment is unknown. I mean, we don't even know what the true mechanism is. So obviously the environment is also kind of unknown. And the problem is that the environment is really important. Like in particular, like in Kimara's previous talk, talking about photosynthesis, the environment there is extremely important in how that photosynthesis is so good in the quantum regime. But in magnetic sensing, it's very underdeveloped. Okay, so that's the model. So what do I mean by now quantum coherence? So quantum coherence is a phenomenon that arises from quantum superpositions. For example, like two states, a state being in two places at once, or a photon having multiple polarizations at the same time. And so this is like extremely important to quantum information. It's basically like basically describes a lot of, it's one of like minimum features you need to have in order to describe quantum phenomenon. And in particular, you can use it as a resource for quantum information tasks. So what I mean by this is that often in quantum information, you can have these games or these tasks that you need to do. And by having coherence, quantum coherence, you do better in your task than if you didn't have quantum coherence. So when it comes down to actually working with quantum coherence, you kind of write states that don't have, you call states that don't have coherence incoherent, and they have this particular diagonal form. So you can write like a quantum state as a density matrix. And so a state that has only terms in the diagonal is called incoherent. And so it means that any state that has terms in the off diagonal are called coherent. So if C and D here are non-zero, then you say the state has coherence. The problem with this is that this depends on basis. So with quantum states in particular, you can always rotate your quantum states and basically change your frame of reference to a basis where the state is diagonal. So basically you can diagonalize any quantum state. So that kind of means that if you have a state and you say that quantum coherence is helping, then what if you just rotate your frame of reference to a basis where there isn't any coherence? Then if coherence doing anything anymore? In fact, yeah, all states have a basis in which they're incoherent. So this is a problem. And how do you get around this? And it's like a problem in quantum biology. And the way we get around this is by using basis independent coherence. So it doesn't depend on basis, and it's in a sense a minimization or maximization over all possible bases. So you say that a state has coherence. If a state has coherence in any basis, then it's basis independent coherence. What this kind of means is that there's only one state that has no coherence in all bases, and that's the maximally mixed noisy state. So it's basically, it has like equal elements along the diagonal and zero everywhere else. And no matter how you rotate this matrix, it will always have the same form. And so in that case, how do you measure basis independent coherence? Because you can't just use the off diagonal terms anymore, because you want to measure coherence in every basis. So instead what you do is use a measure, like a distance measure, and here it's the relative entropy. So basically what you have is your state rho, and then you want to measure a distance, which is like an entropy distance, to the only state that doesn't have any coherence, and that's the maximally mixed state. So from my title, when I say aviance by magnetic sensing, I mean like magnetic sensing in birds, but then we have a simplified model based on what we think it could be, in using like radical pairs and scavenger C, as well as environment collisional interactions. And when I say basis independent coherence, I mean if a state has coherence in any basis, you say it has coherence. So this is how we simulate this kind of thing. So you choose a random state and you evolve it over time. And what you do is to change the angle of the magnetic field. And as you can see, this is like the value here is like the recombination yield. And what happens is that at different angles of the magnetic field, you see you have different values. And the idea is that the bird should be able to sense these differing values and somehow relate the change in the yield into change in the angle of the magnetic field. So what we do is there's some analytical work, but that's in the paper. But here I'll just talk a bit about the numerical work. So in the numerical work, what we do is that we pick random initial states along the Bloch sphere, and then we calculate how each state evolves across all the different angles. So you can see that these lines here is for one state, and as you change the angle, it can change the yield. So this is the initial coherence, and this is the yield. And as you can see that as you increase the initial coherence, these lines become longer. Basically, it means that the change in the yield changes. There's a greater change in the yield over the different angles. And you can see when it's like no coherence, there's basically no change at all. And this is important. So it's important because the change in the yield here is how you're able to detect the change in the magnetic field. So this plot here is basically plotting how big that change is. So as you can see, you have when you have higher initial coherence, you have bigger change in the yield, which means that it should be easier to detect changes in the magnetic field angle. There's different colors here because slightly different states have different react a little bit differently in the evolution, but that's not really important. The whole point is that the trend is important. But there's also another thing as well. Even if the change could be high, it might not be detectable. So if we go back here, we can see that, okay, when you have really high coherence, you have this really great change in the yield. However, what if the bird can't sense anything that's below some value, for example? Like you can't sense like minute changes in the, like you can't sense extremely tiny amounts of the yield, for example. So it's important not only to have a high change, but also to have a high value of the yield itself. Like for example, this value here would be easier to detect than this value down here. So this second plot over here is the product between this, the change versus the actual value of the yield itself. So the idea is that we're kind of balancing, you want high change, but you also want a high value of the yield. And so what we kind of see here is that when you get high coherence, you have a big change, but the value of the yield itself can reach low value, so it's not very detectable. So the idea is that at this kind of midpoint here, it's basically the optimal location. You have a good change in your yield, and the value itself is pretty high, so it should be detectable. And after that, when you add more coherence, it becomes you get diminishing returns basically. So what these kind of results will show is that the key point is that at zero coherence, you have basically nothing. The sensor doesn't work at all. You need to have a little bit of coherence to have a sensor that works okay, like poorly. You have a lot of coherence, and then you get a sensor that works really well. So that's basically the key result that we have, that basis independent is important, it's necessary, and in the particular model that I just showed you, it is probably sufficient. And the reason why it's probably is because it's a conjecture, not a proof. So in summary now, basically what we have is that coherence relative to any basis produces a non-trivial magnetic sensor. Doesn't matter, you can choose a random initial state, as long as it has coherence, doesn't matter what basis it has it in. If it has coherence, then it's useful. And the reason is because the magnetic field keeps changing direction. So as we say, coherence is very useful in a lot of quantum tasks. So, but you can change the basis, and then you can say, okay, now that you change the basis, coherence has disappeared. But with the magnetic field, you can have a state that has coherence in a particular basis. You can say that it is zero in another basis, but the magnetic field can change. So the relative basis has now changed again. So it means that as long as you have coherence in any basis, the magnetic field will eventually go to a basis where that coherence becomes useful. So in quantum biology, the two main things that we're really interested in is characterizing how much coherence or how much quantumness is in that biological system and relating that quantumness to the performance. And so what we've kind of shown is that you can use coherence or basis independent coherence as a measure of quantumness. And we've been able to link coherence directly with performance. And this is kind of more going to the future. So what we have is that we have a model, and the model is, this is our best kind of approximation of what this model could be. And in magnetic sensing, you need to have these things called like extra symmetry breaking in order for the model to work. And this can be done with hyperfine interactions or electron dipolar interactions, or with the environment collisional interactions that we introduced. But the problem is that like we don't know what the environment is. So hopefully in the future, there will be more work to characterize the raw environment. And that's the end of it.
All right, thank you very much, Tao, for a very interesting talk and a very interesting topic. So the floor is now open for questions. Yes, I see one hand by Ilya. Yes.
Hi, thank you very much for interesting talk. But I have I'm a little bit confused. Because I was thinking that, I completely agree that we have very fuzzy idea about, how the ambient magnetic reception works, and it's still highly discussable topic. We don't know, what's happening on kind of how what is the cryptochrome environment kind of in the eye and so on and so forth. But I think that where the consensus was that what is the initial state, and everybody was agreeing that it's a singlet state. And as being an entangled state, they have such a property that they are independent with respect to the LOCC transformation, or kind of the local rotations. So it's really, and I'm not really following where is your initial conditions are coming in, and why it's not a singlet. Or
so the idea is that we're looking at how it actually works in the first place. So the singlet state has entanglement in it. So we want to say, like, if you didn't have entanglement, would the sensor itself still work? And this also means is that if the singlet state has any noise, for example, then it will still work. Basically, our results show that it's like very robust against noise. Yeah.
Okay. But where in that case, where the performance is coming from?
So the performance is just coming from the coherence. Like, it's not the entanglement in the singlet state that is like that leads to the performance. It's the coherence in that singlet state.
Okay, thank you.
Okay, I hope you're satisfied with that answer. Otherwise, you can maybe discuss further after the session. Right, I see there's another hand by Namdi. So please go ahead.
Thank you very much for this topic. Please, I want to know, why is the magnetic sensor needing extra symmetry breaking to work?
Okay, yes. So in the basic model of the magnetic sensor, you have the Zeeman interaction, and you have this integral that integrates over to get the recombination. And in that case, if you have no other interactions, if you only have the magnetic field interaction, it's basically unless you have unless you have something that breaks the symmetry, the magnetic sensor doesn't work. Basically, it evolves exactly the same. Like the recombination is always exactly the same no matter what angle you have. You need to have some kind of anisotropy in order for the change in the angle of the magnetic field to actually have an effect on the recombination yield. Yeah, like the mathematical details is given in the paper that I have.
Thank you.
Okay, right. Thank you very much. Thank you once again, Tao, for the very interesting talk, contributing to our conference. Yes, so let's give her another round of applause. And then the next speaker, the last speaker for this session, will be Dr. Michał Gwizdała, and he's from the University of Pretoria in South Africa, and he's going to talk about phycobilisome's secret life unraveled with single molecule spectroscopy. So while he's setting up, sharing his screen, maybe let me give some justifier of why this talk is in this session and not in this afternoon session, where it would actually fit a bit better. Yeah, well, we didn't want to make this afternoon session too long. And yeah, so this talk does focus a bit less on quantum mechanics than the other three talks, but we could in principle justify this by noting that fluorescence, the process of fluorescence, is a quantum phenomenon. And by investigating these light harvesting complexes at the single molecule or the single protein level, they do behave like quantum units. A lot of interesting quantum mechanics involved there. Right, Michał, the floor is yours.
Okay, thank you. Can you hear me? Because I hear a lot of noise. Okay, I think it's better now. Okay, thank you for the introduction, Chad. And yes, I'm Michał Gwizdała. I work at the University of Pretoria, and I'm going to tell you about our studies of phycobilisomes, studies that involve single molecule spectroscopy. And as it was mentioned, it will be some sort of a smooth transition from quantum biology to molecular biophysics. That's how I see it. Just to also at the beginning I would like to thank the organizers for putting together this meeting and for giving me a chance to present those data to you. And I hope you will enjoy it. So the majority of work that I'm going to talk about was done in South Africa, the University of Pretoria, in the biophysics group led by Professor Chart Kruger. On those projects, I work particularly close with Tesfaye, Joshua, and Farouk. So in this group, we study photosynthesis. And photosynthesis is a molecular mechanism that converts one type of energy into another type of energy. It works a bit like a windmill that converts kinetic energy of wind into potential energy of water. And as by definition, photosynthesis is a biophysical process because it takes place in a biological context, but it's a physical chemical process involving, I will talk about later about some physical aspects of it. Photosynthesis faces some challenges related to input variance. So, variations. So it means that actually the amount of light that photosynthesis gets changes, and also the needs of organisms change over time. So photosynthesis has to have ways to respond to that. As such, in general terms, molecular machinery responsible for photosynthesis can be divided into two components. These are light harvesting complexes that work a bit like the sails or the wings of the windmill. And there are reaction centers that work more like a mechanism, like gears inside the windmill. So they actually do the conversion of one type of energy into another type of energy. In our research, I'm going to focus almost exclusively on light harvesting complexes. And in particular, we've been working with light harvesting complexes from cyanobacteria. Those light harvesting complexes are called phycobilisms, and they are quite complex, as can be seen on this structure model. Because of the complexity, I'm going to use this toy model that shows the functionality. So they absorb light and transfer excitation energy to the reaction center. And as such, they are composed of a large number of pigments that are bound and embedded in protein matrix. And this protein matrix actually is responsible for many of the characteristics of those pigments. So we can even isolate those phycobilisms from cyanobacteria, and we'll see that we get this blue pigment. And when we illuminate them, we can even see with our naked eyes that there's this very, very bright pink fluorescence coming out. When we study phycobilisms in more detail, we'll see that they are composed of, in general, 3 types of compartments. There are those distal rods that transfer energy to the central core, that then transfers energy to the terminal emitters, which are responsible for transferring energy to the reaction centers. So they work like a cascade of energy. And they have been studied for actually around 200 years now. And one way of studying them was actually to just collect steady state emission spectra. And we are not the first people who did it, but we can see this nice broad peak of emission. In our studies, what we wanted to understand, what we wanted to know is what's behind this broad peak, whether there's any dynamics, whether there are any heterogeneities there. And for this reason, we decided to use single molecule spectroscopy and look behind those broad peaks. Single molecule spectroscopy allowed us to avoid averaging over sample concentration and observe heterogeneities of emission, or detect some rare states if there are any present. So how is it done? How are we able to look at get a signal from one complex? So we start with isolated sample and we dilute it by a lot. And then we deposit this diluted sample on a glass slide, and we expect phycobilisms or the complexes to bind to the surface of the glass. Then we scan the glass with laser beam, and once we hit a phycobilism, we can detect fluorescence. For this, we're using our home-built custom single molecule setup in Pretoria. It's based on a confocal microscope principle. So once we hit our complex, we can actually, we know its location, we can come back there and we can measure different parameters of this emission. We can measure spectra, intensity traces, and lifetimes of emission. And we can measure even more, but in this process, in this project, we are actually focusing on all those three. So now let's go to the results. Here in gray, you can see how the intensity of emission changes over time. And what you can clearly see is that it's not constant. So in bulk emission, you have a straight line. Here you have some sudden, let's say, switches to different, less bright emissive states. We can fit those this intensity traces with those blue levels of intensity, and we can then resolve the lifetimes of emission for each single level. And if we look at the correlation of lifetimes versus intensity, what we can see that they are linearly correlated. So that tells us that those deep, less bright states are due to quenching process taking place close to the terminal emitters of the phycoblissum. What this correlation also tells us is the relative size of the complex. So the slope of this correlation will inform us whether our complexes didn't, for example, fall apart during the measurement. And this is a high risk in our case. So we are able to analyze the switches already in that way. And we know that the switching between the states is due to quenching mechanisms. So we wanted to look more into details at the switching between different states. And first we use low light intensities, and we could see that there are not that many switches between different states. However, when we increase the excitation light intensity, we would observe that there are far more switches between different states. We could quantify them and look at the switching rates from unquenched to the quenched state and from quenched to unquenched state. And you can see that with increasing light intensity, indeed, both those switching rates, switching frequencies increase. What's also interesting here is, and you can see it's quite unique for single molecule studies, we were able to use light intensities that are much lower than a physiological limit of amount of light that photosynthetic organisms can get in nature. So we were able to show that actually the switching, those dynamics take place also in under conditions that are similar to the natural conditions. But what's most interesting is when we compare how those switching rates, switching frequencies change. And what we'll see is that actually with increasing light intensities, phycoblissums are able to shift the equilibrium between the states towards the quenched states. What it means is that when there is too much light coming into the system, phycoblissum will dissipate it before, for example, in the cells, it could reach the reaction center, or when isolated, before it would be seen as fluorescence. So we basically demonstrated here a new energy dissipative mechanism for phycoblissums that is light activated, directly light activated. We wanted to know what's the origin of those switches between different states and entering this quenched state. And for this, we used the glutaraldehyde, which is a chemical cross-linker, and what we expected to create some sort of a scaffold around the protein matrix and hinder any conformational changes. And what we observed with increasing amount of glutaraldehyde is that the switching rates in both directions actually decreased. This shows that conformational changes are involved in the switching between different states. So the protein matrix is not only holding pigments in place, but it also regulates its functionality. So then we also look at the emission spectra. And here you have plotted in how the fluorescence peak position changes in time. And if you look at those green and blue region, you can see that the shift in fluorescence peak position is correlated with quenched state. And indeed, if you look at the spectra, you can see that there is some blue shift here. To understand what it means, Chart developed a kinetic model in which phycoblissum was divided into a few compartments, and we assigned a quencher to different compartments. And this model is shown here as a line, as the lines. So then when we plotted our data points, what we observed is that actually the quenching, so those switching between different states, can take place in any compartment of a phycoblissum. So each part of a phycoblissum is capable of entering a state in which it dissipates energy. Most likely this happens in those red compartments here in the core or in the terminal emitter. Then we also observed another type of spectrum that was very atypical, and this is something that we didn't expect. So we observed that some complexes could switch and enter states with a characteristic additional red band in the emission spectrum. And we were intrigued about that because this is something that you cannot as such see in bulk spectroscopy. And we also wanted to understand it better. So first we look at the correlation of those red states with quench states. So it turned out that typically those red states or far red states actually take place just after or just before the quench state. So indicating that they are correlated. Then we look at the dwell time. So for how long a complex can stay in this with this additional red, far red emissive band. And it shows that actually there's a very stable band. So this is not an artifact of a method. What's somehow also interesting, very interesting actually, is that those far red states are also light induced. So they are as light induced as those fully quenched states that we observed previously and discussed previously. That also gives us an idea that they are involved in those quenched states somehow. Then we compared the bulk spectrum of emission here in black with the sum of all single molecule spectra here in blue, and you can see that they're identical, and that's okay. But then when we took only the fully unquenched spectra, we observed here in red that there is less contribution on this red side. And this tells us two things. It tells us, one, that the red states are present in bulk as well. So they are contributing to this bulk spectrum that we observe, that we observe, everyone observes, everyone knows. And also that the red states are, again, correlated with the quench states. To understand the character of those red states, we use Stark spectroscopy. It's a method that allows to detect directly charge transfer states. And we relatively quickly realized that we always need to include a red band around 705 nanometers in order to be able to analyze the data. And as it turned out, this red band has a strong charge transfer character. That tells us that those far red states have a strong charge transfer character. Then we had a base understanding of physical-chemical properties of phycobilisomes, how they interact, how they behave when they are alone, isolated, alone, and illuminated. But we wanted to, since this is a biophysical project, we wanted to do something more physiologically relevant, something with more biological sense. So we decided that we'll try to reconstitute in vitro a physiological photoprotective mechanism that involves orange carotenoid protein. And a few years ago, I reconstituted this mechanism in vitro in bulk, and it involves isolated orange carotenoid protein that is orange. Then this protein can get photoactivated and it turns red. And this red form can bind to phycobilisome and actually quench the fluorescence from a phycobilisome. So we wanted to repeat this in single molecule spectroscopy. To do this, we started again with our phycobilisomes bound to the surface of the glass. We added orange carotenoid protein. At this point, nothing was happening. We shined laser on the phycobilisome. We could see emission. At this point, nothing special was happening. But then when we added additional source of blue light, the light that could photoactivate OCP and turn it into this red form, we are expecting to observe quenching of emission. And this is how our results look like. So here you have a trace again of fluorescence emission.
You have 2 minutes left.
Okay, thanks. We could observe fluorescence emission. And when we shine the blue light, we will observe deep quench state. And this was irreversible because we know that OCP when bound to phycobilisome is reversible. So we just were able to observe a deep quench state that lasted for a very, very long time. This was very exciting, but this was not satisfying because we know that in nature, using other complexes, OCP mechanism is reversible. So we used OCP isolated from a mutant that has low affinity to phycobilisomes. And here we were able to observe that a reversible quenching of phycobilisome at the level of 2 individual physiological partners. We were able to see that those quench states induced by OCP are much more quenched than the blinking related states that I discussed before. But what's most interesting is that we saw intermediate states of quenching, and this is uniquely available for single molecule spectroscopy. And those intermediate states have very, very particular spectrum. First of all, it has a red component here that indicates that OCP induces charge transfer states in the phycoblism, but also the blue component is blue shifted in comparison to other spectra. What it tells us is that when OCP binds to the phycoblism complex, it actually decreases the connectivity in the phycoblism complex. So this allowed us to create suggest a new model, a more detailed model of interaction between OCP and phycoblism, in which OCP, photoactivated OCP, needs to decrease the coupling inside the phycoblism in order to fully quench it. So far I was talking about quench states. Now I would like to mention very briefly another type of state that is not quenched. So here we only look at the rods of phycoblism. And to our surprise, in single molecule spectroscopy, only 70% of the rods had a bulk-like spectrum. And some of the rods actually were able to switch between the bulk-like spectrum and another state that was far-red, that had a spectrum that was far-red shifted. We were able to see that switching between the state is also driven by conformational changes. But what's most surprising is that the red state is not quenched. So far, all the red states that I was discussing before were quenched. So they were not really bright, they were not emissive. Here, it was a bright state that, well, from a physiological perspective, that would mean that this could function as a light harvesting complex. And indeed, we identified a paper which showed that those rods alone, so not the whole phycoblism complex, could bind to a photosystem 1 and serve as a light harvesting complex. And when we compare now the overlap between emission of phycocyanin and absorption of photosystem 1, we can see that the red form that we identified, that we discovered, is actually has a 40% increased overlap with the absorption of photosystem 1. So that tells us that phycoblisms, this form could serve as a light harvesting form for photosystem 1. So to conclude, phycoblisms do have hidden dynamics. There's a lot happening behind those broad peaks. Typically conformational changes are involved in those dynamics. Some states have charge transfer character, something that was not known before. And we were able also to see physiologically relevant mechanisms using SMS. And that in general, this flexibility of emission allows phycobility proteins to play different parts in photosynthetic apparatus. I should thank at the end our collaborators in France, Netherlands, Bangladesh, our group again, and the funding that we got to do this research. And thank you for your attention.
Right, thank you very much, Michał, for an illuminating talk on a topic that's obviously very interesting to me. So the floor is open for questions. Okay, any questions are welcome, even if they are very basic ones.
I do have a basic question. So you do these experiments on this lab bench that you have in the picture here?
Yes. Some of the experiments that I showed were done here, yeah.
Okay, so then in your labs, you have the ability to prepare the samples in your lab, or do you work with like a chemistry or biochemistry group?
Until now, we are working with the laboratory where I did my PhD, and this is a laboratory in France, in CA-Saclay. So we are isolating phycoblisms with them. Now we're working on a small project to actually have a Nigerian collaborator to help us also isolating phycoblisms, but this is not, it doesn't work as yet. And we also think about doing it in Pretoria at one point.
Okay.
Right, thank you for the question, Lawrence. Maybe a short question from my side. So Michał's single molecule spectroscopy is typically done in environments that are very atypical from the physiological environment. You can maybe comment a bit on that and why you would say that these results have physiological relevance if they are done in such an artificial environment.
Yeah, that's actually a very good point, and this is very often the criticism that we face when we for example publish a paper. We get reviews that say this is very artificial. From the chemical perspective or biochemical perspective, these conditions are not different than any other in vitro studies of light harvesting complexes, and this is a widespread way of analyzing light harvesting complexes, simply to isolate them from the cells and to look at them in vitro. So when it comes to this, we are not far different. We are actually no way different. We're the same. When it comes to light intensities that we use indeed in SMS, very often light intensities used are very high. We're talking like a couple of thousand of suns if we compare it like the excitation rate. Here we managed to use extremely low light intensity. So this is again another point against our research that that this is not physiological. We are actually able to push it to physiological regimes. What's most important, I think, is that we are studying with SMS the most basic physical-chemical properties of the pigments that are embedded in protein matrix. And this will not change whether we are in the cells or whether we are in vitro. The properties are simply there. It's just the maybe probability of entering different states is different in vitro or in vivo, but the possibility or the capability of entering those states is the same. It's so both in vitro and in vivo complexes have the same repertoire of states that they can enter. And finally, one of the projects that I briefly described, the interaction with orange curtain protein, that is a fully physiological process. So this is also very unique research in a sense that we took two physiological partners. We didn't do anything to them, so we didn't tag them. We basically isolate them and put them into the device, and we measured their interaction. This is as much as physiological as it can get for any in vitro studies.
Thank you, Miha, for a very clear and elaborate reply. Namdi, would you like to ask a question? I see your mic is on.
Yeah, explanation once again. I would like to find out, is it only the fluorescent detector that can perform this experiment, or is there any other detectors that you can use to do this experiment?
So currently, it's fluorescence that is it's used for single molecule spectroscopy because there's no background and the signal is relatively easy to distinguish from the excitation light. In the past, there were some attempts to do absorption spectroscopy on single molecule level, but this is not done on biomolecules. This is done on some specific chemicals. And as far as I know, it's at very low temperatures. My feeling is that there will be more attempts to do for example, scattering measurements at single molecule level, but I don't think this was done as for now.
Okay, thank you.
Okay, thank you. Any last pressing question before we close the session? Okay, if not, this has been a very inspiring session, and let's give all the presenters of this session another round of applause, a virtual applause. So thank you very much to all the speakers of this session.
Thank you.
Right, so this closes this session on quantum biology, and I would like to remind you about the open forum that's starting in a bit less than an hour's time at 12:30 South African time. And at this session, the African Physical Society and some of our other co-conveners are going to discuss the African strategy for physics, and also some exciting research facilities and opportunities. So please do come and participate in that forum, share your ideas, listen to some other people's ideas about how we can jointly raise, especially biophysics research and biophysics infrastructure on the African continent. Right, and then the next scientific session will be this afternoon on molecular biophysics, which is going to start at 3 o'clock South African time. And a range of very interesting talks are going to be given on mostly biomolecules, so please do not miss that. And thank you very much for joining this session. We hope to see you then at the rest of the conference. So goodbye, everyone, for now.